welcome to my first video on IB psychology. Today we're going to talk about the biological perspective. And my name is Linnea Karlberg. As with anything in the IB, I think it's always important to refer back to the guide. What does the IB actually require us to know? This unit is called the brain and the behavior, and I recommend that you pause right now and read through before we get started. We're going to look at brain scanning techniques, some evaluation, as well as empirical research. Before we get started, there are two concepts that I want to cover. First of all, there's temporal resolution. What does this mean? Well, it's the precision of a measurement with respect to time. So simply, how quickly can the neuroimaging technique measure brain activity? Secondly, there's spatial resolution. It refers to the smallest measurement that a scanner can detect. Think about it like a YouTube video. You want the quality to be as good as possible so you can see every detail, every pixel clearly. So think about this when you're doing your evaluation. The first example is magnetic resonance imaging. This produces a highly static image of the brain. This is done using radio frequency fields, which manipulate the alignment of the atoms. This then causes a rotation magnetic field, which is detectable inside this scanner. Each tissue in the body has different magnetic properties, and this causes contrast between the tissues. This then scans slices, which are assembled using a computer. Evaluating this, we can identify some strengths. For instance, this can be used on any body part, and it's often used for tumors where X-ray may be a concern. Furthermore, it's non-invasive. However, we cannot see brain activity with MRI, we can only see the structure. Also, the exposure to magnetism, it can be dangerous for some individuals. For instance, if you have a screw in your leg after surgery. It also can trigger claustrophobia for some participants. And any slight movement will affect the image. Overall, this technique is quite expensive. Looking at a study where MRI was used, we can talk about Moguire et al. From 2000. He looked into the functions of the hippocampus in spatial memory through a natural experiment. So he was interested in whether changes could be detected in the brains of London taxi drivers. So 16 healthy, right-handed male London taxi drivers were compared to 50 healthy, right-handed gender-matched controls, people who didn't drive taxis. The amount of years for driving taxis were also recorded. Through the uses of MRI, they measured the size of the hippocampus, and this part of the brain has been associated with memory prior. It was found that spatial maps and memory alters brain with expertise. So the anterior part of the hippocampus was bigger in the controls, and that's the front, and the posterior part, the back of the hippocampus, was bigger in the taxi drivers. So hippocampal volume of the right hippocampus in each taxi driver correlated with the amount of time spent as a taxi driver, positively in the right posterior and negatively in the right anterior hippocampus. So a larger posterior hippocampi meant a more proficient at spatial memory and navigation. This then connects to neuroplasticity, which we'll talk about later on. This study was high in ecological validity, as it examined a real-life event. There were also a control group present and clear variables stated. In a later version of the experiment, bus driver was added, and the same result was found. Thus, triangulation provides strength for the study. However, there may be some confounding variables present, and the sample was quite small. Also, since there is a manipulation of the independent variable, we cannot identify a cause and effect. Secondly, we can look at fMRI. This uses a large magnet which measures changes in blood flow to different active areas of the brain. So when neurons in particular regions are active, more blood is sent to that region. This then maps changes in the brain's metabolism to show physiological structures and action as they function. This is also a non-invasive way to record brain signals without risk of radiation, which can be found in other scanning methods. It also has a really high spatial resolution that we talked about earlier. This can record signals from all regions of the brain, It is also widely used and standard in data analysis. It produces a compelling 3D image of brain activation and is relatively easy to use. However, the images produced must be interpreted carefully because correlation does not imply causality, and brain processes are complex and often not localized. This also has a poor temporal resolution because the bold response peaks out approximately 5 seconds after neural firing begins in an area. A study where fMRI was used is by Passamonti and Crockett in 2012. They looked into the role of serotonin in regulating emotion in the brain. This was a laboratory experiment, and there were two conditions. There was one serotonin depletion group. They were given a mixture of amino acids that lack tryptophan, which is a building block for serotonin. This group had little serotonin. The placebo group was given the same mixture with a normal amount of tryptophan. Afterwards, the volunteers' brains were scanned using fMRI, as they viewed faces with angry, sad, and neutral expressions. They then measured how different brain regions reacted and communicated with another. Furthermore, a questionnaire was distributed to determine if the participants had a natural tendency to behave aggressively. Low brain serotonin made communication between the amygdala and the frontal lobes weaker, and this was even more pronounced in individuals with a natural tendency to aggression. So the researcher concluded that low serotonin levels lead to more difficult for the prefrontal cortex to control emotional response to anger that are generated within the amygdala. This study had a control condition, which is really great, a good strength. Also, clear correlation was found, and through the fMRI, precise and robust results was produced. It was further comprehension of serotonin's role in emotional processing. However, the study has low ecological validity, because when will you be inside an fMRI scan looking at facial expressions? Also, some individuals cannot have fMRI scan, and that limits their generalizability of their results. Another way of studying the brain is EEG. This is when electrodes are placed on the outside of a person's head in specific locations. This then detects changes in electrical activity below them in the form of action potentials or nerve impulses. These are then detected by electrodes and graphed over a period of time, as you can see here. And this is then thought to reflect the processing time for brain's communication and information processing. The best thing about the EEG is that the hardware costs are significantly lower than the previous discussed option. It also allows for mobility. You can move around and do studies about movement. It also enables a higher temporal resolution. However, furthermore, it's silent, so it's better to use when studying responses to auditory stimuli. And it doesn't aggravate claustrophobia. However, it has lower spatial resolution compared to the fMRI, and it's not sufficient for localization of function because it detects an uncountable number of neurons on the surface of the skull. Another way is PET scans. This involves the injection of radioactive dye, which is mixed with glucose, into participants. The dye then produces measurable gamma rays as the sugar is metabolized in the brain. More metabolization of glucose that occurs in a given brain region is inferred to mean greater activity. This then detects the rays and turns them into computer images of brain activity. Brain activity is then color-coded by software based on the amount of glucose metabolized in a specific regions of the brain. PET offers the potential to identify a variety of diseases and illnesses in their earliest stages on a cellular level. It is also useful for showing abnormalities in brain activity levels in diseases that don't really show structural changes until much later. An example of that will be Alzheimer's disease. Important to note is that the injection doesn't stay radioactive for long, so the risk from radiation is very low. It can also be used to track ongoing activity in the brain. However, is it really ethical? The injection may hurt and it's prone to allergies. Furthermore, PET is expensive and time-consuming, so you cannot do any longitudinal studies. Okay, let's look overall at the techniques we looked into today. What are their strengths? Well, through this brain image technology, we can look into healthy, functional human brains without any real fear of damage or change. It is also way less invasive than another option, which would be exploratory surgery. We're able to establish broad correlational evidence between a given behavior in a brain region, and this can then be further researched and triangulated. Also, early diagnosis and treatment of disorders like autism, Alzheimer's disease, and schizophrenia can be done. It helps with identifying the brain areas that involve in problems like, phantom limb pain. And yeah, overall it can help overcome pain. However, there are some limitations as well. Firstly, it's unlikely that the brain imaging technology can reduce any given behavior to a single neuron or molecule. Human behavior results from an interaction of multiple biological, cognitive, and social processes that we're going to talk about more in this course. Also, it's important to account for the environmental influences. Simply, there's a lack of precision relying on software to interpret the measurements. Errors could occur without the researcher's knowledge, and that reduces the validity. Many of these also cause stress and claustrophobia, and there's a lack of ecological validity in the studies. Also, since these techniques are expensive, it reduces the total number of participants and the likelihood of replication. Since not so many people can access these technologies, it's only the wealthier people who perform the scans, and does that make the results generalizable? Definitely something to question. There are also some ethical problems associated with the sensitive data that comes upon after doing these technologies. There's also a problem of interpretation, since many of these technologies involved a lot of colors, can give the impression of more change than there actually has been. And something that's super important to know is correlation does not something very important to note is correlation does not equal causation. That's all for today. If you have any questions, please comment down below and we'll get more into psychology in the next video.